Slide. Okay. Okay, thank you very much for this great opportunity. To the organizers, it's a privilege and honor to be here to talk a few minutes about our research at bioengineering department. So in June of 2022, the US Congress passed an act which was called FDA Modernization Act. This act, as you see here, and you can look it up online, authorized FDA to basically look for alternative solutions and alternative platform technologies to spare animals and bring safer and better treatments for our patient and bring down the cost of drugs. So the question comes up, what is the rationale and what was the justification to pass this act? As most of us are aware, the process of drug development is a very lengthy process for pharma and basically academic labs, and also it's very costly. It takes on average 2.5 billion and about 10 to 12 years to get a drug from the basic research to the patient. And then many of these drugs that are actually in preclinical trials are failing because there's a lack of predictability of current preclinical models to show the efficacy of the drug. So before they get to the clinical trials, they kind of fail. And by that time, the pharma and the others, they have spent a lot of money on these and put a lot of resources. So there's a notion on drug development says fail early and fail cheaply, basically. So what are the preclinical models that we use mostly in our labs or pharma use to do drug basically testing? So a lot, as we know, animals are gold standards for a lot of foundational biological studies to see how a disease spreads inside the body, such as cancer, and also to test numerous compounds in the form of single kind of like assays or combinatorial treatment, and etc. Despite significant advancements that these preclinical models, animal models have made in our discoveries, there are significant challenges. So the challenges are, animals are not human. The physiology and the pathophysiology are different, basically. And then there are numerous confounding and complex factors. So it's almost impossible to dissect the role of a single component, such as a blood vessel, or a single cell type, such as an immune cell, T cell, or just macrophages or fibroblast cells, on finding out what are the biological bases that disease kind of spreads inside the body. So all these factors come together and dissection is going to be difficult. These are expensive and time consuming. I was in the NIH grant study section. There was one R01 for 5 years. They wanted to sacrifice 1,000 mice for one study in New York City. I shouldn't say that, but also the animal rights in a very expensive area. The animal rights that we don't want to talk about it, but that's a huge kind of discussion right now going on across different groups. So that actually justifies the need to develop better preclinical and predictive assays that we can enhance the efficacy of drug screening and drug discoveries, actually. So the question comes, can we develop these assays to replace the animal models or complement? So I don't want to say completely replace because that's a bold statement. To complement the animal models and basically enhance our preclinical discoveries. So since 2010, there has been an emergence of a field called organ on a chip, tissue on chip technology, although I personally believe that organ on a chip was existing before 2010, but it was rebranded as a new name when it was introduced to the scientific community. So organ on a chip or tissue on chip technologies integrate different tools that we have had as bioengineers or as engineers and even biologists from the past. Those are micro and nanoscale technologies that most of us are aware. For instance, semiconductor was there from years before to make microchips. Biomaterials such as hydrogel, synthetic and natural base and natural biomaterials, and also the biological assays that we use, standard biological assays, and also the cells coming from the patients, learnings from medicine, and etc. So we can integrate these tools to develop biochips or ex vivo chips that we can go directly from the patient to the chip and make our basically studies in a very well-controlled environment and dissected manner. So this is the schematic of one of the projects that we worked on since joining ASU about 2014, was in collaboration with Mayo Clinic in Arizona and Josh Lab and Jay is here. So we could take cells from the patients in Mayo Clinic of Arizona from breast cancer patients of different subtypes. Take the biopsies back to the lab, digest the cells, and then design a microchip and then put the cells inside the microchip so we can replicate the tumor of the patients outside of the body. So what you see on these biochips, so they can distribute it now. I think I have brought some of these chips so you can look them around. On these biochips, as you see the size of these chips, so we have a nice tumor entity. So we can add tumor cells in any hydrogel that you want, such as collagen, matrigel, hyaluronic acid, and etc. We can have a stromal layer. There are guideposts that we have designated to create a nice organotypic entity to separate these two entities while they are interconnected to each other. Now we can add immune cells here, we can add T cells, macrophages, and we can remove them. We can add a drug compound or biochemical factors. Pretty much these chips are compatible with any biological assays that are existing, and you can conduct studies in a well-controlled manner. Now let's zoom into these chips. So what you see on this blue dot box is what's happening actually here. These are tumor cells which are loaded into the tumor region of the chip. The dimension here is just 1 millimeter, basically, and then this one is 2 millimeters. So we loaded the tumor cell. These are the guideposts, and then we stimulated the cells with epidermal growth factor, and then let the cells to go out. So we can look at a single cancer cell. We measured the speed of a single cancer cell in X, Y, Z, and see how these cells kind of maneuver throughout the 3D tissue using this technology. And even we can go further down. These are the microposts, 100 microns, single size of a hair. And this is the cancer cell, brain tumor cell, which are extending to the brain kind of capillaries or blood vessels. And this work was done in collaboration with Barrow Neurological Institute, that they're one of the leads in brain tumor kind of like discoveries and preclinical trials. So we are going into the blood vessels and capillaries, and we can look at single cell junction using this technology. So these technologies actually provide a lot of capabilities for us to kind of do our assays and preclinical drug screening and foundational biological studies. They're organotypic. It provides 3D high-resolution imaging. It's compatible with a lot of biological assays and molecular kind of like assays, such as single-cell RNA sequencing, Western blotting, qPCR, and etc. Now we have developed different chips. So we have tumor on a chip, breast tumor and brain tumor. We have Alzheimer's disease on a chip, and we have heart attack on a chip. So this was done in 2020. Recently published in Science Advances in 2022, and we mimicked cardiac diseases on a chip, such as heart attack, lung QT syndrome, and heart failure with preserved ejection fraction, which there is no cure for that disease. And we identified novel compounds which lead to the disease progression. So this is the chip on this basically microscope stage, and we are shining a fluorescent light into the chip, and we can take biopsies from the patient. These are stem cells beating inside the chip. We can take biopsy from the patient who has a heart disease, reprogram back to the stem cells, and then now program forward to the cardiac cells. And then we can have cardiac fibroblast cells or beating cardiomyocytes. We can co-culture them, mix them, look at beating frequency. We can add drugs, and we can do so many compounds screening on this to basically see what are the what causes to the disease progression. So I'll stop here. I think it was just 7 minutes, but as I mentioned, these technologies have a lot of impact on the community moving forward. It brings a nice synergy between clinician, foundational fundamental biologists, and also bioengineers. And then we have, right now, we are at the verge of starting our first company to commercialize this chip and make them user-friendly. So we can make them very accessible for different labs that they can run their assays. Thank you very much.